Hey everyone watching, I'm Katie.
I'm Virginia.
I'm Mary.
And welcome to our informative video presentation on cells. Let's start the conversation off on important structures of the cell. Plasma membrane. What's a cell? When most people look up a picture, common computerized images appear to make it look like a circular, egg-like figure with squiggly stuff floating about. Believe it or not, cells have some similarities to your typical chicken egg. Who knew? Both the egg and individual cell have a membrane that allows certain molecules to leave and enter while also acting as a barrier for foreign molecules, pathogens, and overall maintain a homeostatic environment.
When looking at the structure of a plasma membrane, you can see these linked-looking things with heads and little tails. These are known as lipids, and they make up the phosphodilayer, bilayer meaning 2 layers of lipids. The heads face either end of the barrier while the tails, properly known as fatty acids, face inward on either side, forming the nonpolar region. Typically, there are 3 membrane lipids that contribute: phospholipids, glycolipids, and cholesterol. These can attach to the heads or the base of the membrane and are able to change the charge and structure.
Another important factor in the cell membrane are membrane-bound proteins. 2 noticeable proteins are peripheral proteins and integral proteins. Much like their names, peripheral proteins are loosely located on the rim of the membrane, while integral proteins are located through the membrane and aid in the transportation of molecules in and out of the cell body. There are 6 main functions of membrane proteins.
Let's do a countdown!
Number 1, transport. As we touched on before, proteins assist with transport, meaning allowing certain molecules to enter through its channel, such as integral protein. Number 2, enzymes. All enzymes are proteins, but not all proteins are enzymes. Enzymes break molecules into another form. For example, it can break apart a lactose molecule into 2 monosaccharides, being glucose and galactose. Number 3, receptors. Protein receptors implant themselves into the rim of cell membrane. They can relay secondary information to the cell by having a chemical messenger, like a hormone, attached to the protein and send its translated message through the wall. Once the message is received, a response is then activated inside the cell. Number 4, intercellular adhesion. This is used in cells for communication and to receive/distribute materials or water from one another. This can occur with the involvement of gap junctions and the process of tight junctions, but we'll touch on these later. Number 5, cell-to-cell recognition. There's millions of cells floating around in our bloodstream. It must be easy for a foreign body to sneak by, right? Well, the cells in your body are actually marked with proteins called glycoproteins, which help cells identify each other and/or individual cells. Number 6, attachment to cytoskeleton and extracellular matrix. Proteins located in the cytoskeleton of a cell help to maintain the cell's shape, while proteins in the extracellular matrix, or the ECM, assist in intracellular and extracellular exchanges of the cell. Between the lipids and proteins that make up the cell membrane, there's a third, carbohydrates, more specifically, glycocalyx. Carbs can also link onto lipids and proteins, resulting in glycolipids and glycoproteins, which assist with cell recognition, signaling, and being able to fight back against invading pathogens. Microvilli and membrane junctions. Some other features of membrane of the cell are the microvilli and membrane junctions. Microvilli are on the surface of the membrane of most of your cells in your body. People sometimes get them confused with cilia because both of them are finger-like extensions of the cell. Cilia are actually assist with cell movement throughout the body, while microvilli are only used for absorption of nutrients and secretion. That's why the largest amount of villi is located in the intestinal tract of your body, to help absorb more nutrients and important fibers to fuel your everyday bodily functions. There are 3 main junctions that contribute to intercellular adhesion and to cell-to-cell contact: tight junctions, desmosomes, and gap junctions. Tight junctions create a watertight connection between cell membranes, while gap junctions form tunnels that allow molecules to pass. Desmosomes, on the other hand, commonly attach to the protein keratin, which is why you can normally find them in your skin. They tightly hold cells together and are a major factor in why your skin doesn't just roll off. Besides the structure of the cell membrane and the types of proteins and other features that make up its surface, one of the most important jobs a membrane has is being able to let certain materials in and out of the cell body. This occurs with the process of selective permeability and diffusion. Selective permeability does exactly what it sounds. It selects certain molecules or ions to enter or leave while permitting unknown particles from entering the membrane. With diffusion, there's three main types: facilitated diffusion, simple diffusion, and osmosis. Facilitated diffusion is the movement of hydrophilic molecules or ions across the plasma membrane. Simple diffusion is when ions or molecules diffuse from an area of high concentration to an area of low concentration. Finally, in osmosis, water molecules move from a region of low solute concentration to a region of high solute concentration across a semi-permeable membrane. Moving on from osmosis, we have tonicity, which determines the shape and change in a cell by measuring the water volume. There are three different classes of tonicity for solutions: hypotonic, which has a high water concentration level with a cell, causing it to eventually burst; hypertonic, that has a very low water level within its cells, causing it to shrivel; and isotonic, which has an equal amount of water inside the cells.
The cytoplasm and organelles of the cell. Next, inward past the membrane, is the cytoplasm, which fills the entirety of the cell. Cytosol is the liquid component that makes up the cytoplasm within a cell. It also carries the various organelles that help a cell function. What are the organelles? First, we have the nucleus, the largest organelle that you can find in all types of cells. The nucleus, it has some very vital roles, so let's come back to that one later. Next, we have the mitochondria. You might have heard that the mitochondria is the powerhouse of the cell from Mr. Bill Nye, but this may be true, but I'd put this down as an answer that you'll most definitely get wrong. Most specifically, the mitochondria produces ATP, or also known as adenosine triphosphate, which carries the chemical energy that the mitochondria produces for biochemical reactions throughout the cell.
Next up on our list are ribosomes. Both free ribosomes and bound ribosomes synthesize proteins within the cell. Although bound ribosomes synthesize proteins for export, exiting the cell, and lysosome usage. These are typically found on the rims of the rough endoplasmic reticulum, while free ribosomes are found floating around among the other organelles. Which brings us to the endoplasmic reticulum, or also known as the ER. The two types of ER, rough and smooth. Remember our friends the ribosomes? Well, similar to the bound ribosomes, rough ER contributes to protein producing and sending them away in vesicles. Vesicles may fuse with organelles such as rough ER and travel through the cytoplasm to transport proteins throughout the cell. The second type, smooth ER, is different from rough ER because instead of producing proteins, it produces lipids. Smooth ER is also known to help with detoxification of waste, and why cells found in the liver have a high amount of smooth ER. You can identify the two because the rough ER has ribosomes in it, and is usually surrounding the nucleus, while the smooth ER is usually tube-like and does not contain any ribosomes.
Next organelle is the Golgi Apparatus, which is the factory of the cell. The Golgi Apparatus receives, modifies, and sends out packaged proteins. Enzymes in the Golgi Apparatus contribute to modifying the molecules and sorts of the materials that are sent. The Golgi Apparatus can be seen with vesicles or lysosomes coming and leaving it.
Moving on to lysosomes, you can normally see them as little round spheres moving about the cell body. Lysosomes are filled with digestive enzymes and are involved with various bodily functions such as breaking down older parts of the cell and dissolving invading pathogens. Traveling to either end of the cell, you'll find these star-shaped organelles called centrosomes. And making those up are centrioles. The function of the centriole is to arrange the spindle fibers in a mitotic cell division. The centrosome's function is also centered around cell division by organizing preparing the centrioles for cell division. On the very outer rim of the cell is either cilia or flagella, also commonly known to confuse people. Both assist in cell movement but look entirely different. Flagella has a tail-like appendage that helps the cell swim along in any direction, commonly found in male sex cells, while cilia are hair-like follicles located all over the outer rim of the cell membrane. And last but not least, one of the most vital organelles, the nucleus. The nucleus itself is known to hold your genetic material, as well as controlling the actions that happen within the cell. On the rim of the nucleus is the nuclear envelope. The nuclear envelope, also known as the nuclear membrane, is made up of two lipid bilayer membranes that in eukaryotic cells surround the nucleus. The nucleus encases your genetic material. The nuclear envelope consists of two lipid bilayer membranes, an inner nuclear membrane and an outer nuclear membrane. Inside the nucleus is the nucleolus, where ribosomes are produced. Nucleoplasm fills the inside of the nucleus to support its shape and plays a vital role in transporting materials necessary for cell metabolism and functioning. Finally, there's chromatins. Chromatin is the material that makes up a chromosome that consists of DNA and protein. Its super important function is to tightly pack DNA into a cell. Cell life cycle and protein synthesis. There are three main parts of a cell's life that it must go through: interphase, cell division, or scientifically known as mitosis, and cytokinesis. First up in the batting cage is interphase. This can also be broken down into three parts, being G1, S, and G2. Initially in G1 phase, the cell basically lives up to its normal life, physically, and it happens to increase the volume of both protein and organelles. While in S phase, the cell copies its DNA to produce two sister chromatids and replicates its nucleosomes. Finally, G2 phase involves further cell growth and organization of cellular contents.
Next is mitosis. This is when a cell divides and goes through 4 different stages: prophase, metaphase, anaphase, and telophase. Essentially, this is how those stages take place.
In prophase, chromatin condenses down into chromosomes. The nuclear envelope then breaks down. Chromosomes attach to spindle fibers by their centromeres. Moving on to metaphase, chromosomes can be seen lining up along the metaphase plate, or the center of the cell. In anaphase, the sister chromatids are pulled to opposite poles of the cell. In telophase, nuclear envelope reforms and the chromosomes unfold into chromatin. Finally, there's cytokinesis. Cytokinesis is a physical process that finally splits the parent cell into 2 identical daughter cells. During cytokinesis, the cell membrane pinches in at the cell equator, forming a cleft called the cleavage furrow. Moving on to protein synthesis and RNA. DNA is copied into RNA during a process called transcription, and by which RNA is used to produce proteins that aid in another process called translation. RNA plays a major role with its 3 types: mRNA, tRNA, and rRNA. The main difference among mRNA, tRNA, and rRNA is that mRNA carries the coding instructions of an amino acid sequence of a protein, while tRNA carries a specific amino acid to the ribosome to form the polypeptide chain. And lastly, rRNA is associated with proteins to form ribosomes.
Thanks for watching, guys. Don't forget to smash that like button, hit the subscribe, and click the bell icon for more notifications.
And that sums up our presentation of the cells.
Fly, be free.